Thank you. My name is Dr. Komozo Makaola. I'm the Senior Science Manager with African Society of Laboratory Medicine. It is our great pleasure to welcome you to this session. For those that are returning, we are grateful for your loyalty. And for the new ones, I do believe you will learn something and you will be part of this journey going forward. As laboratory professionals in Africa, we do a lot. In a day, we generate tons and tons of data, and we also have millions of samples that pass through our hands. And through the ASLM Science webinar series, our intention is to capacitate laboratory professionals to go beyond just issuing of results, to go beyond the daily taking care of patients, but to be more intentional in using the data, the information that we have in an ethical way to develop our profession. There is a great opportunity in science to innovate. There is great opportunity to change the way we do things. Hence, we established this science webinar series. And just for your information, we do have translations in French. We also have translation in Portuguese. So you can select the channel where you would be able to listen and understand in a language that you understand better. So through this science webinar series, like I've said, we want to enhance the skills of laboratory professionals in research and its dissemination, to also equip laboratory professionals with the knowledge and utility of emerging technologies and innovations in diagnostic medicines. I do believe today we have amongst us policymakers. So this is an appeal, a call to them to show them the different areas, the different components of laboratory medicine that exist, so that as they program, they are able to incorporate these in their new strategies. We also have amongst us the upcoming laboratory professionals. I know we have a lot of students that are in attendance, and we just want to say to them, beyond probably what you are seeing, your routine hematology, your routine microbiology, there's a lot more that you can do within laboratory profession. Hence, today we are going to be talking about molecular and precision medicine. And allow me to welcome our speaker, Dr. Goodwell Nzo, who is a molecular biologist, a translational scientist, and educator with over a decade of experience at the forefront of precision medicine, molecular neuroscience, and disease modeling. Dr. Goodwell is originally from Zimbabwe, but he's now in the US, and he brings a multidisciplinary and global perspective to biomedical science that is shaped by formal training in molecular medicine, neurobiology, and biotechnology. He holds a PhD in molecular medicine and translational sciences with advanced certifications from Wake Forest University School of Medicine and the Wake Forest Institute for Regenerative Medicine. He also completed postdoctoral training in neurodegenerative disease. I think he will pronounce these words much better than me. Research at the University of Pennsylvania, and he has led pioneering work on the development of complex human relevant in vitro models, such as brain on a chip, liver on a chip, lung on a chip. So he's really learned. In both academic and industry settings, Dr. Nzo has led leadership roles advancing microphysiological systems and organ-on-chip platforms for pharmaceutical screening and disease modeling. He's passionate about democratizing biomedical innovation, and Dr. Nzo focuses on developing ethical, cost-effective models that are applicable under resource-limited settings. He has taught and mentored students and professionals across Africa, Asia, Europe, and North America, emphasizing critical thinking, laboratory profession, and translational relevance. He's a strong advocate for interdisciplinary collaboration, and Dr. Nzo continues to shape the field of precision medicine by building scalable, human-centered models that accelerate discovery and improve patient care worldwide. So, ladies and gentlemen, let us welcome Dr. Nzo as he will be speaking to us on molecular and precision medicine. Over to you, Doctor.
Thank you, Dr. McCullough. So today I'll be talking about molecular and precision medicine, but as I was waiting to come on here to speak, I started to think about a quote from an ancient novelist, Charles Dickens, in his book, A Tale of Two Cities. And in that book, the first line of that book says, "It was the best of time, and it was the worst of time. It was the spring of hope, and it was a winter of despair." And as I think of the topic that we're going to be discussing today in terms of the innovations that we've had in the past 30 years, going back to the sequencing of the human genome that was published in 2003, to the founding of Yamanaka Factors for Stem Cell Research in 2006, and to organoid research, bioprinting, CRISPR-Cas9 for gene editing, as well as now we're talking about AI. As a scientist or anybody in medicine, if you're thinking about all of those innovations, you could say these are the best of times, right? But then on the flip side of that, there are patients whose complex diseases, such as complex cancers or neurodegenerative diseases, those people have no treatments that are available for them. And for them, it is the worst of times. So my hope is that this conversation that we're going to be having today will be able to help you guys see how scientists are working towards really removing that distance between the best of times and the worst of times, where these innovations are actually being used to bring together the bench closer to the bedside. So in talking about molecular medicine today, I will be sharing with you some definitions in the field. And I will be talking a little bit about where we started in terms of what we have learned from traditional medicine or traditional models as to what we have gotten from that in developing precision medicine tools. And then we'll go into the details of the advanced model systems, which in some cases are called microphysiological systems or organ-on-a-chip. There are different terminologies that are being used in the field, but I will be using the advanced model system as for nomenclature in this conversation. And then, of course, I would like to show you examples where these platforms are actually being used to respond to problems or challenging issues that are in the clinic. So without further ado, the definition of molecular medicine is that molecular medicine seeks to understand a disease at a molecular level to inform diagnostics as well as therapeutics, which I simplify as the molecular medicine asks what's going wrong at a cellular level. And precision medicine is about tailoring treatment to the individual variability in genes, environment, as well as lifestyle. So, which I also like to term right treatment for the right patient at the right time. And when we're thinking about all these terms in terms of molecular medicine, there are other topics that starts to appear: genomics, proteomics, and bioinformatics. And there are multidisciplinary approaches that are developing tools to drive personalizable treatments. And some of these include single-cell epigenome, where you're focusing on resolving the cell state and lineages, and looking at the epigenetic editing, where you're focusing mainly on the precision control via looking at CRISPR-Cas9 modifiers that are being used now to understand specific epigenetic editing. And then also looking at chromatin profiling, that's also used in molecular medicine to study histone marks and DNA accessibility, and other tools that are also being used right now in molecular medicine to understand genomics and proteomics. Is also looking at epitranscriptomic mapping, where you're profiling RNA methylation, as well as now AI, of course, where everybody's talking about multi-omics integration, where you're finding new patterns as well as biomarkers for treatment. And going back a little bit in terms of the definition that I mentioned earlier, molecular medicine asks what's going wrong at the cellular level, and precision medicine is using that answer to decide what treatment that works best for that particular patient. And in going back to the existing models that have been used in molecular medicine, we have learned quite a bit from these traditional models: mouse models, rats, rabbits, and monkeys, and many, many other types of non-human models. We have learned and established protocols, and because of that, the regulatory agencies accept the protocols that we have been able to develop from this. And you are also able to study complex systemic responses in animals, unlike what you might be able to do in a cellular model. And for 2D models that are simpler, that have been used in the past 50, 60 years, those are easy to scale, and also they are cost effective. And the idea that you have the availability of actually genetically modifying an animal that actually helps you to look at a specific disease in an animal system. With all these advantages, there are also challenges that actually come with these systems. As you can imagine, a rat, a mouse, a rabbit, or monkey is not a human. So you can imagine that there is a limited human relevance where these animal models do not have the physiology that really mimic human physiology. And there are many, many ethical concerns that are related to animal use and welfare. And the 2D systems that are being used, the 2D simple models, they do not mimic human physiology or the 3D structure that you would expect in a human being. And as many scientists or many, many clinicians that might be on the call, you might be aware that there are over 95% of the drugs that are failing clinical trial, not mainly because these drugs are just horrible drugs, but sometimes it might be because of the models from which these drugs are developed that's making them not succeed in the clinical trials. And many, many scientists believe this. And over the years, scientists have been developing new technologies to respond to those issues, to those challenges. And in the context of molecular and precision medicine, this is how scientists came up with the advanced model systems, which are innovative, biologically relevant platforms that closely mimic human physiology and disease condition in order to actually accurately predict therapeutic efficacy, safety, as well as mechanism of action for particular disease pathways. So what are these advanced model systems? They range from simple single cells, which are iPSCs, to many, many complexities where we are looking at a body on a chip. We have iPSCs, organoids, organ on a chip, bioprinted constructs, and body on a chip, where you're connecting many, many, many organ on chips together to look at the proper physiologies. But just to highlight a few here, the organoids are miniaturized, self-organized 3D tissues that are derived from stem cells that mimic real organ tissue like brain, liver, or lung, or intestine. Sometimes people are using primary cells from cancer patients to make organoids so that they can mimic that physiological conditions of that particular cancer of the patient. And then organ on a chip, these are devices that use microfluidic technology to really recreate the physical as well as the functional dynamics of a human organ. For example, fluid flow to mimic blood flow, and then also having mechanical forces to mimic specific organ tissue types. For example, if you're talking about in the context of a lung, you might want to mimic the breathing motion of the lung, or if you're trying to mimic the intestine, you might want to mimic that peristalsis that you would expect to see in the intestine, so that you expect the cells to express proper genes with which you can do proper experiments. And because of this developments that scientists have been working on in the past 20 years, the regulatory agencies are also jumping on this because they are seeing the value of these platforms. As you can see here, there are so many regulatory agencies across globe that are actually promoting non-animal methods for drug discovery, as well as also for preclinical testing. And I'm very pleased to say that in Africa, I see a few countries that are also adopting these technologies. For example, South Africa is really working on and is interested in incorporating the 3D in vitro models and organ-on-chips technologies for their preclinical models. And this continues as we see with the adoption from the regulatory agencies. We're also seeing the private sector jumping in on this. There are so many companies right now across the globe that are developing platforms that are in line with these non-animal model use. For example, the organoids, there are so many other companies, iPSC platforms, AI tools, organ-on-chip, as well as body-on-a-chip. There's so many other companies. And if you go online, you can find the specifics or the specific applications that some of these companies are looking at. But to jump into how these advanced model systems are playing a role in precision medicine, I just want to show you here with iPSCs, for example, you are able to genetically characterize iPSCs from a patient that has a specific condition. For example, a patient that might have neurological disorders, for example, Alzheimer's disease, Huntington's disease, or Parkinson's disease. And there you can actually precisely model that specific disease phenotype at a molecular level, with the idea that the interpretation of the data from that system really mimics what you might expect for a particular patient. And with organoids, because you're extracting cells from the patient and then making these micro tissues, you are able to represent the real person because you're taking that real person's cells so that you have a person's real tissues. And scientists across the globe are also making biobanks where they are extracting, they're making organoids from people of different ages, different backgrounds, and different health conditions. This helps us to make new treatment, not just for one person, but also for many, many others. Because if you are only working with one cell type from one patient, you might have a treatment for that particular patient. But with these biobanks, it actually gives us a picture as to how we could develop treatments that work for a lot of other people. So because of these organoids that are being used, that are being extracted from patients, you can imagine they help us to find the right medicine for the right patient, and also to understand how the body might react if you're looking at a toxicological assessment using those particular organoids, as you will see in some of the cases that I'll be sharing with you. And in looking at organ-on-chips, these are microfluidic platforms that I've seen for quite a while now, but really the idea is almost the same, that there are multiple compartments. Some of them have 2 compartments, some of them have 4 compartments or 3 compartments, but the idea is to really mimic that physiological structure of the target tissue or target organ. For example, if you're trying to target the neurovascular unit, you might want to have the vasculature to mimic the blood vessel, and having the parenchymal channel that mimics the actual brain tissue itself, and having the right cells that are separated by either the physical membrane, and sometimes they use hydrogels to mimic the barrier between the 2 tissue components. But these microfluidic organ-on-chips are actually enabling the mimicking of proper human physiology by really having these mechanical cues as well as fluid flow. And we know, for example, in the lung, the breathing motion of the lung actually helps the differentiation of the epithelial cells and also to express specific markers that are necessary for the epithelial cells to function the way they're supposed to. And then the fluid flow, of course, there are some organ-on-chips, there are some tissues, for example, the vasculature, where the maturation of the endothelial cells, or even the expression of the specific markers within those cells, depends on that fluid flow. So this is what really the microfluidic systems are really trying to address. And because of that, you're able to do personalized drug testing because you can do toxicity screening of therapeutics. You can do dynamic functional analysis because, for example, they might incorporate gold MEAs onto that chip, so you can pick up the electrical signals. For example, if you're looking at heart cells or heart organoids, if you're looking at neurons, you could also use these platforms for understanding the synaptic firing of the neurons. So this complexity go further than that. I was talking about the organ chips where you can only focus on one organ chip, but there are other platforms also that are trying to incorporate multiple organs together so that you can have the full complexity of a body on a chip where you can assess multiple effects. But in this case here, these body on a chip platforms are actually helping to personalize drug testing, where you can predict how a drug responds in metabolism or toxicity, and also reducing the trial and error that you would normally have. For example, a patient who's presenting with a specific disease, but they might not have a proper treatment for them, but there might be 5 options. But you can have this in vitro twin where you can screen the drugs to see how a drug would react in one organ tissue type. And if there are any subsequent effects for metabolism, then you can actually assess that and have a clue as to what's the way forward. And in summary of all these advanced model systems as to how they are playing a role in molecular medicine and precision medicine, how they are pushing this field forward, is that you can see how iPSCs, because of being able to do mutations in specific cell types, you can actually have this genomic application to understand how that particular cell would behave because of that mutation. Organoids can be used to understand the 3D tissue proteomics, also even looking at drug responses and biomarkers that you could expect for a particular application. And organoid chips, for example, you can look at drug effect or drug effect prediction at an organ level to simulate the specific organ tissue type based on what drug you're looking at and also what disease model you're looking at. And then the body on a chip, I consider this a digital twin model that you can have if you're looking at under bioinformatics, where you can have a twin model of that body on a chip, where you have all these cross interactions that you can map based on the data that you are extracting from the body on a chip. So now I will share with you a few examples of how these platforms are being used in the clinic as well as in preclinical settings. So, for example, the iPSCs are being used in finding methods or finding drugs that work for patients that have very, very complex diseases. For example, in this particular case, this is a case that was for an 18-year-old patient who had an ultra rarely high mitochondrial disorder, whose iPSCs were derived from fibroblasts, and then they tested across multiple drugs in vitro. And then they identified 3 top drugs from that screen, and then these drugs were administered to the patient, and within 3 years, the metabolic profile for that particular patient shifted to normal. And you can see how these platforms are actually being used here for personalized as well as targeted therapy to help the patient, not just tinkering around at the bench, but also translating the bench to bedside. And here is another case. This was a phase 1, phase 2 trial, which really was dependent on deriving iPSCs, derived dopaminergic neurons that were transplanted into 7 patients with Parkinson's disease. And at the primary endpoints of this trial in particular, they were focused mainly in the adverse effect, of course, and they saw no tumor formation and there were no serious adverse effect. And for their secondary endpoint, they saw dopamine production via PET, of course, and then they looked at the motor improvement for all the 6 patients that were in the trial, and then they noticed that 4 out of those 6 patients had motor improvement. And you can see how iPSCs are being used not just for rare diseases, but also for neurodegenerative diseases that are now prevalent in many, many societies. And now I'll move over to showing you how patient-derived organoids are being used in cancer treatment. There are two cases that I'll be sharing here. This one is a case of 126 patients that had sarcoma, and that they formed a biobank of 110 organoids from these patients. And what they did, they made from those organoids, they were able to do a high throughput screening of those organoids to see what drug would work best for these patients. And these patients had their sarcomas were not responding to any therapies. And so after those screens, they identified at least one drug that worked for over 59% of these cases. And they were able to match the drug response in the lab with the organoids with the real clinical outcome. And so you can see here how these patient-derived organoids were working as a predictive tool to understand how you can actually get an effective drug that works for the patient. And this is another trial of cancer trial where they were looking at 90 patients that had metastatic colorectal cancer patients. They had their tumors-derived organoids that were generated from biopsies of 44 patients with no satisfactory treatment choice. And they did another drug sensitivity test in these tumor-derived organoids, which pointed out to specific treatment choices for 34 patients of these 44 patients that they had made the organoids from. And they were able to see 50% disease stabilization of the 34 patients that they treated with those medications that they identified, which actually helped them to meet the primary endpoint for progression as well as the free survival for that particular trial. Again, this showed that these patient-derived organoids can successfully guide real-world therapy, not just being used at the bench. And now what I would do is I'll share with you some of the data that is actually being generated in the field with scientists in preclinical settings, where scientists are using these models to understand how drugs interact in a more complex system. And this study in particular was a study that I worked on with my colleagues at Wake Forest, where we incorporated 6 different organoids onto the chips. They were all connected together with 2 setup, where with liver, intestine, lung, brain, as well as testes on the chip. And then that was one setup. Another setup, we removed the liver to understand what would happen in a case where a drug that gets metabolized by the liver and the metabolite might have an effect. So we looked at specifically with we looked at ifosfamide, which is an anti-cancer drug. This is a chemotherapy that is known to have a lot of side effects in patients. So we're trying to see if we would be able to capture this on this platform. So with this, in the setup that had the liver, the drug was metabolized, and we saw subsequent effects in the brain model, for example, where we saw toxicity with also the inflammatory cytokines that were being expressed in the brain organoids. And that actually showed that this drug is getting metabolized by the liver, and there's drug-to-drug interaction that's actually, there's an organ-to-organ interaction that's taking place within the entire system. And then when we did not have the liver, there was no effect of that drug in particular. And the question that you might ask, especially if you're talking about in the context of Africa, that are these platforms applicable for infectious disease? Yes. Here is an example of a platform that was developed to understand the evolution of an influenza virus. And here they used a lung airway chip, where there are two compartments, with the vascular compartment with endothelial cells and then the parenchymal compartment with the lung epithelial cells. and the epithelial cells were exposed to air. They did not have any fluid, just like what you would expect in the lung. They had mechanical forces to mimic the breathing motion of the lung, and they were flowing medium with the specific nutrients within the medium. And they were able to really track down these virus mutation in the airway chip. But what they did here is they treated the chip, they introduced the virus into the airway chip, and then to mimic human-to-human transmission, they did a serial passaging that you can see here in the bottom picture, where from passage 1 to passage 2, there was a transfer of the extract from the top compartment into the next chip, and then repeating that 8 times to see how a virus is responding to a drug from passage 1 to passage 8. And as you can see in the figure here, that at passage 1, the drug is the virus is actually responding to that drug. But by passage 8, you have this mutation possibly that's causing the resistance of this virus not to respond to this particular drug. And they were able to really see these natural selection pressures that are normally seen in human lungs. And they were able to see the emergence of mutant virus strains within these conditions that were also similar to the strains that they would expect in the clinic. And this platform is actually being used now to provide insight into drug resistance as well as viral adaptation mechanisms of these of any particular viral infections. So you might say, what's the significance of this? Well, this demonstrates that organ-on-a-chip technology can predict clinically relevant viral mutations, and it also offers a preclinical tool for evaluating vaccine resistance, as well as optimizing antiviral strategies. And also in the context of pandemic preparedness, it can help the health surveillance, as well as organizations that are responsible for making sure that they understand how a virus that might start in a small community, how it might end up affecting other communities by having these lung models that could be made from different sources of cells. And in conclusion, I want to say that as we have seen today, advanced model system, whether they might be iPSCs, organoids, organ-on-chips, these are not just scientific novelties or innovations, but they are the future of medicine. And they're helping us personalized medicine, personalized treatment, and predict outcomes. And looking at beyond animals, the shift from animals, really, many people might say is only ethical, but I think it's also practical, because these systems are helping us reduce failure rates in clinical trials and offer faster and even safer path to patient care. And then if you're looking at the real world impact, patient-derived systems are transforming how we actually understand and even treat diseases, from rare disorders to complex cancers, as you have seen in some of those examples that I've shared with you today. And in conclusion here, I want to say that now our challenge and our responsibility, and I'm glad to see that globally, everybody is focusing on how we can actually transition towards using models that really mimic human physiology. And I really hope that we can continue to invest as well to advance these models, because I believe that the future of medicine is not just about innovation or coming up with new tools, but it's about delivering healing that is personal, predictive, and equitable. And with that, I thank you.
That was a mouthful. Thank you very much, Dr. Nzo, for such a very informative lecture. At this time, I would be opening up for questions, but we will start with those that are in the chat, in the Q&A panel. Dr. Nzo, I hope you are able to see those. The first one says, could there be any of these innovations, like the organoids, at phase 3 or at marketization phase?
I have seen I've seen these models being applied at phase 2, phase 3, where a drug that might have been in the clinic for a little bit, and then they saw toxicity in a different organ, and then they want to evaluate that at a molecular level to see the mechanism of action for that secondary organ chip. For example, a cancer therapy that might be targeting lung cancer, but then they see toxicity in the kidney, and then developing a kidney model to look at that toxicity. I've seen those applications, but again, I'm not sure if it's prevalent yet. But these applications, they work at a different levels of I would say the drug pipeline. It could be in the phase 4 or even monitoring itself, right? It really would be able to work for many, many stages, I believe.
Thank you very much for the response. And others are mainly just congratulatory messages. And there was a question about whether this presentation will be shared. It will be shared together with other documents. And there is also a question about, are there distance online learning programs available for molecular diagnostics? You can also combine it with the one that says, what is your take between MSc in biochemistry and pharmaceutical science in terms of the future of medicine and teaching institutions? Is there software for these models?
A software for the models. Software, I think now with the AI, a lot of people are considering integrating these models into those AI tools, where, for example, just understanding what data you can collect from the system, and how to parse out the markers that you're getting from the models. For example, in a human body on a chip model, for example, you could be looking at biomarkers from the lung model, biomarkers from the liver model. How you make sense of the data that's coming from all of those, I think that's where bioinformatics comes into question. As far as incorporating MSCs in molecular medicine, I think this is really where it really started, where when people are looking at MSCs and looking at stem cells, that's how it really started, where people are starting to see the opportunity to use personalized cells or cells from an individual, or developing MSCs that you could, differentiate into a specific lineage for your particular application. And this is really what really made these advanced model systems work the way they do now, because we have learned quite a bit in terms of the molecular insights from those platforms.
Thank you very much. The other question is, how can resource-constrained countries like Zambia begin to implement personalized medicine approaches using the existence healthcare infrastructure? I think basically the question is, what you were presenting, is it feasible in resource-limited countries?
So I believe that it's doable, especially if you're only looking at the iPSCs as well as the organoids. If we remove ourselves from the more complex microfluidic platforms, those that might need equipment or secondary translation of the data that's coming out of that. The organoids, if anyone that has worked with cells, should be able to work to develop organoids, because as I mentioned, the organoids, they self-assemble into small tissues. And it's just a matter of having a proper plate that actually where you could grow these organoids, and then the interpretation of that data from there depends on the general molecular biology experience that you might have. But yes, it is doable in a resource starved environments, I believe.
Thank you. Thank you very much. I'm trying to scan through. I think most of these are also, is this technology available to be developed in developing countries? Is it only possible in more developed? I think you have touched on that. Is there an explanation for the lack of response in real life versus molecular laboratory generated therapies?
Yes. So there
Yeah,
is
that one.
yeah. So for that, I would say it's mainly the standardization of these model systems, because for the past 15 to 20 years, scientists have been developing the models in a different way. What I might characterize as a brain model might not be the same brain model that someone else in the lab B is developing. So I feel like as more regular agencies are really supporting these developments, they will emerge a standard that might actually make it universal, so that the adoption as well as the universal use will become standard as well.
Thank you very much. The other one is on opportunities. Do we have any online trainings for molecular diagnostics? And then do we have any PhD scholarships to study molecular medicine?
So I would say most universities in the United States have programs that are looking at some of these organ systems. But as far as scholarships, if you get accepted into a PhD program in the United States, I believe that most of them are PhD funded, and for small short-term training courses, there are conferences that you could go to. For example, there's a World MPS conference that happens sometimes in June, I believe. And then the Workforce Institute for Regenerative Medicine runs a course every summer for people across the globe. I've seen, when I was there, we were training scientists from different parts of the world that were coming in for a week to learn as much as they could in terms of whatever they wanted to focus. If they wanted to focus just on organoids or organoid shape, or just learning on the theory of the specific areas, there are all those courses. But I do believe that large universities in the United States have similar programs or courses online that they are also offering for people to learn about this. But generally speaking, there are many presentations that anyone that is interested in this field could look up and find many well-trained scientists across the globe that are talking about this.
Thank you very much. I see maybe the last one was how can AI be used to provide personalized medication?
So AI comes in at the intersection of molecular medicine and bioinformatics. And the reason I discussed it in the sense that it's a downstream effect of molecular medicine is because you're actually collecting the data from a cell with which you are using to interpret how to treat a patient. But the focus of this presentation was really to highlight the environment in which the cell is, which determines what genes are being expressed, as well as what proteins are being expressed as well. So that was the discussion. But again, I think there are many AI tools that are being used right now to interpret the data. If you're looking at omics, for example, that is really coming in with together with AI as well as bioinformatics.
Thank you very much. I realize there's a number of people asking about certificates. Please note that certificates will be issued once this session. There will be an email sent to you about when you can download your email, your certificates. So those details, we'll share them together with the material. And there was another question. I don't know if there's anybody with a burning question that wants to open their mic, and I can allow for 2 questions, oral questions. If there's anybody that wants to ask, you can show by raise of hand. Okay. I don't see anybody. The last one that I'm going to read, it says, there's a hand. Please unmute yourself and go ahead.
Thank you so much for this opportunity. I just wanted to know, if we had to set up something like this in an African country, what would be the cost in terms of doing just the organoids? We have to push this through a government system.
Yeah.
I think it's a brilliant talk, but I'd like to know what it will cost and how we can motivate for this type of practice, because I think it's the way the future is going to go. Thanks.
Yeah, I would not know the exact number, but the general discussion I would have with you on that is if you already have a cell culture system or a cell culture modeling that you're already working on, you with the organoids, you should not have to invest a lot more in terms of the incorporating the organoids into your workflow, because it's about the cell sourcing, right? If you are able to purchase the cells, most of the cell types from different vendors, they are not very expensive, or relatively they are affordable. So I would say just the source of cells and media, and then maybe the place in which you can culture those organoids. It should be less than $10,000 just for that particular setup. But if you're trying to do multiple tissue types, multiple applications, then it depends. But that number is just a general number that I'm giving you without actually knowledge of how much it might actually cost, depending on where you're ordering the cells from.
Thank you so much.
Thank you, Prakash, for that question. We have Ahmed Uma. Please go ahead and ask your question.
My question is, when using organoid, it is important to use for resistance pathogen because of there is a gene mutations and another problem during antimicrobial resistance. How to make solution for this antimicrobial resistance by replacing this organ replacement? Thank you.
Dr. McCullough, maybe you can help me in terms of understanding, Ahmed. The way I understood that is, how can you do how can you do anti-resistance, drug resistance studies on organoids? Is that the question?
Yeah.
Okay. So with the example that I showed with the influenza virus, where you are using the organoid chip to understand drug resistance, you can do that. It's a similar method for the organoids as well, where you can have multiple organoids that are cultured, and if you are looking at drug resistance for a virus that's going into the organoid, you could take the supernatant from one culture, transfer it to the next, to see how over time, if the virus is changing or a specific bacteria is changing in any way, or even the response to a specific drug is taking place. So the setup is much simpler in the organoids than it is in the organoid chip, for sure, but it's doable.
Thank you very much. I will read maybe the last round. Anonymous says, I feel these advanced methods are not well known in some places of the world. So how can we make it accessible and known to the world at large? You presented some, I don't know if it's the same person. Maybe it's helpful if you include your names. Please go ahead. Go ahead, Dr. Nzulu.
Yeah. So I would say, in terms of making it well known across the world, it's one of the efforts as well when I reached out to you to talk about this, is because I really believe that these platforms are very useful in preclinical as well as in clinical settings. So conversations like this and going to conferences, and since the launch of the World MPS program, I think that conference, it has been expanding quite a bit. I remember the first meeting, I think it only had about 2,000 or under 1,000 people that attended. But the third meeting that happened, or fourth meeting that happened this year, had more than 6,000 people, or even more than that, actually, I think. So as those conferences are growing, and there are other smaller versions of those meetings where people are talking about advanced model systems, I think it's just a matter of exposing ourselves to the conversations around the world. And for those that are trained in these technologies, talking about it and sharing the ideas, and going to universities that are having conferences and presenting the data and sharing that, and or even methods that others are using. So I think those are the ways through which we could help others across the world to know as well as understand how these technologies are working.
Thank you very much. So that's, like we have summarized, it's really one of the objectives why we hold these sessions, because we've realized in some parts of the world these technologies are not well known, while in other parts they are used almost every day. And the last question that I'm going to take, it says, could you please speak to graduate training in molecular biology versus health informatics versus bioinformatics? Which is more relevant to this field of molecular medicine as a laboratory scientist? You could speak to the downsides of either route for somebody focusing on research and academia. So I believe it's somebody who is interested in professional development.
So I would answer that with it, as long as you are in life sciences, it really is up to the student that ends up with a PI or with a principal investigator and suggesting a project for yourself that actually incorporates how to do all of these things. A good example is that for myself, I went into the Institute for Regenerative Medicine, where the focus on bioprinting as well as other platforms. But I went in there to focus on molecular medicine and how to link these complex models with molecular medicine, where I was looking at the pathways of molecules going into the brain and developing the actual brain model. So it really depends on what project you end up doing. But the program under which you are does not necessarily matter, and unless if the university does not have the say any applications that they are working on, or there are no professors on site that are actually working on those projects, then it might be a difficult thing. But if you are going to a university that has some form of advanced model systems that they are working on, you could go into a molecular medicine department or bioinformatics department, but then end up working with a professor that's working with these models. Your project might be developing these models, but then with a bioinformatics application to it.
Thank you very much. So basically, it would depend on the program that you select in terms of theory, and then also your project. You can be intentional to combine these health informatics and bioinformatics and molecular biology in your project so that you get the most out of them. But because in most institutions, you will find that there is a network for the different projects. So you can have a little bit of everything, and then by the time you finish, you have gained everything. The very last one is their phase 4 or 3 study on efficacy or toxicity. And then, has any of these been done in the African continent? Have we used any of these? Yeah.
Phase 3, phase 4, I have not seen it. But I think slowly they will be incorporated into the phase 4 for monitoring any drug that might be already out there. Has any of these platforms being used in Africa? As I showed you with the example, I believe it's on this slide here, where you have these African countries that are already moving towards these non-animal models. And so, yes, as you see, these specific agencies in these countries are pushing for this, then you can imagine that there are some universities that are now incorporating this. You might not see clinical trials that are being run on this yet, but I imagine if there's a clinical trial that is being run by an organization here, depending on the size of the clinical trial, they might want a specific patients from the African continent, but it might not exactly be the trial that's being run primarily in Africa. But I see this probably going towards that direction in the next few years.
Thank you very much. I think we've come to the end of our webinar, and it has been very informative. It has raised a lot of questions in our minds, and that is really the objective, to say, open a window to say this is what is available, and to encourage you to go on. We will be sharing the slides with some links. Somebody was asking about the different conferences. We will put in some links there. Some training institutions. If we find any institution that is training on the same area, we will put them. But the encouragement is, you've been made aware, go and explore. For those that are still going through educational development, and then for the policymakers, you also look at what is it that you can adopt and bring services closer, more personalized services to your patients. With those, thank you very much to our IT, our interpreters, and our presenter, and to the audience. Bye-bye, and see you next time.